Everyone. In this lecture, we're going to be exploring the cell. In all of the previous lectures, we've talked about various forms of living organisms. And so in this particular video, in this particular lecture, we're going to talk about the building blocks of those organisms, which are cells. To begin with, let's take a look at a little bit of the history of the cell and how it was discovered. In 1665, we credit Robert Hooke with being the first scientist to take a look at cells. In fact, he gave them the name cell because he looked at cork cells when he was looking at cork underneath a microscope, which is what you see right here on your right. They reminded him of the cells of a honeycomb, and so he named them cells. But he didn't realize he was looking actually at the cells of plants. Then Anton Leeuwenhoek was one of the first to describe a living cell under a microscope, and this would have been about 10 or so years later. There were other scientists who contributed to what we refer to as cell theory, and we'll see exactly what cell theory is here in a minute. Matthias Schleiden discovered that plants were made of cells. Theodor Schwann concluded that all living things are made of cells, and in fact, we have Schwann cells, which we've named after him. And then Rudolf Virchow concluded that all cells come from other cells, and together these are actually all part of what we refer to as cell theory. That the cell is the basic unit of life. All living things either are cells or they are made of cells, and then all cells come from other living cells. So these are the three parts of what we refer to as cell theory. So here's a typical animal cell. And at the end of the classification video, I gave you a good bit of information about animal cells, and so you can take a look at all of the different organelles here. From lysosomes, ribosomes, the cilia on the outside right here, the cell membrane, which all cells have a cell membrane that encloses the cell membrane that encloses the cell. It's what keeps what's supposed to be inside inside and what's supposed to be outside outside, so that there's a separation between the living cell and the environment. And so that's the cell membrane. We have smooth and rough endoplasmic reticulum. We have mitochondria, we have Golgi apparatus, we have peroxisomes and lysosomes and secretory vesicles and whatnot, centrioles. So we have all of these different organelles that are inside this animal cell. Now remember, what was the term that we use for a type of, for the whole domain of organisms of cells that have a nucleus and membrane-bound organelles? That would be eukaryotes, right? Eukaryotic cells, because we're in domain eukarya. Here's a typical plant cell. And as you can see, most of the same organelles are here, but we also have a large central vacuole. Vacuole is a storage area inside a cell. And in a plant cell, that large central vacuole is primarily responsible for maintaining the water pressure inside the cell. Plant cells also have a couple of other things that animal cells do not have. A cell wall. Animal cells do not have a cell wall. The cell wall of a plant cell is made of cellulose, which is a complex carbohydrate. And then chloroplasts. Chloroplasts are responsible for being able to take energy from the sun and convert that into chemical energy, storing it in the form of glucose. And that would be that whole autotrophic part of a plant. So we have two basic types of cells, prokaryotes and eukaryotes. Now if you'll notice, these are, I've actually spelled it with a C here on this slide, and that's because depending on where you look, you may find eukaryotic and prokaryotic spelled with a K or a C. And it depends on where you find it. Excuse me, and whether it was written in a European book or in a North American book and whatnot like that. So all cells can be separated into prokaryotic and eukaryotic. Prokaryotes contain all of the eubacteria, which would be domain bacteria, and all of the archaeans, which would be domain archaea. Eukaryotic cells would be all of those things that have that are in domain eukarya, with plants, animals, fungi, and then all of those organisms that up until now or up until not now, but some 20 years ago really, were referred to as protists. Here's some basic differences that I do need for you to know about prokaryotes versus eukaryotes. Prokaryotic cells are the oldest form of life, dating back some 3.5 billion years. They have no nucleus. They have very few organelles. They only have ribosomes, actually. They are very small. Most of them are about the size of a mitochondrion or a chloroplast, which, as we talked about in the previous video on classification, that according to endosymbiotic theory, eukaryotic cells actually evolved from symbiotic relationships between prokaryotic cells. Eukaryotic cells evolved a good bit later. In other words, prokaryotes evolved on Earth first. Then eukaryotes evolved from those prokaryotic cells. Eukaryotes have a nucleus where all of their genetic material is contained within the nucleus, surrounded by a membrane. Plus, they have other membrane-bound organelles. They're all part of what's referred to as the endomembrane system. That's because the membrane that surrounds them is exactly the same kind of membrane that surrounds the cell itself. It's made of the same phospholipid bilayer that makes up the cell membrane. And eukaryotic cells are much larger than prokaryotic cells. Here's a typical bacterium. As you can see, it doesn't have any nucleus, but all of its DNA is in a circular DNA that tend to be found closer to the center, and we refer to that kind of as a nucleoid. They do have cell walls. Now, the cell wall of a bacterium is not cellulose like a plant, and it's not chitin like a fungus. It's actually something called peptidoglycan, which is another complex carbohydrate that actually has some elements of proteins inside of it as well. And you can see some bacteria will have long flagella, and they'll have these cilia that allow them to move and whatnot. Most bacteria are motile. There are some that are sessile, but most will move around. Here's a typical animal cell. We've looked at that before. So cells are responsible for maintaining something called homeostasis. Homeostasis is a balance between the inside and the outside. That cell membrane is extremely important for this because the cell membrane controls what gets into the cell and also what leaves the cell. And that's responsible for maintaining this balance, the homeostasis within the cell. So the definition of homeostasis, maintaining a constant balanced internal environment, and the cell membrane is responsible for that. So if we think about how things can move in and out of a cell, you have something called diffusion, which is when things move from an area of high concentration to low concentration. Osmosis is the diffusion of water, and things are simply moving down their concentration gradient. Some things can move directly through the cell membrane, other things can't. The cell membrane, being that it's a phospholipid bilayer, the outside of which is, if we think about like an Oreo with the two pieces of cookie and then the cream in between, the cookie is what's referred to as hydrophilic, meaning that it will attract water and it can, it is able to be dissolved in water and that sort of stuff. The cream filling of an Oreo, that's like the lipid tails of a phospholipid that are kind of intertwined with each other in between the two cookies. That creates a hydrophobic layer. So that prevents anything that is water soluble is prevented from simply moving through the cell membrane. Which is good because, like I said, the cell membrane keeps what's inside and what's outside. It gives you this compartmentalization, which was responsible for life itself, because you have this little compartment that contains all of the chemistry that's going on for life, and then outside of it is the outside environment. Water can move through the cell membrane, but it does have to have these channels that are made of proteins called aquaporins. And so water can go into and out of a cell that way, and that helps maintain this balance. So it's kind of like a door, but it's a door that doesn't have a doesn't have an actual door that opens and closes. It's just an opening, and only water goes into and out of it. And so water will easily move into a cell or out of a cell depending on where the concentration is greatest. Or actually where the concentration of solutes is the greatest. If you've got more solutes inside the cell, then water is going to come into the cell. And if you've got more solutes outside the cell, then water will leave the cell. And that's what I'm showing you right here. If we have a semi-permeable membrane, and we have a high concentration of solutes on one side of it and a low concentration of solutes on the other, over time, those solutes are going to move through the membrane if the membrane will allow that solute to go through. Okay, and this is simply diffusion. So this is diffusion of a solute that can go through the membrane. And so this is just another illustration of that. Now, if we think about solute not being able to move through a membrane, but that water can, then water molecules will move from an area of high concentration to low concentration. In this case, the sugar molecules can't go through the membrane, but the water molecules can. Well, if you look here on this side, the left, you'll see that there's more water molecules here than there are on the right-hand side of this membrane. So these water molecules are going to move across this membrane, and so what you'll end up with is what you see here on the right-hand side, so that you have an equal concentration of water molecules on both sides of the membrane. But what that causes is this side then rises and this side drops. And this happens whenever we put a cell into some different kinds of solution. If I put a cell in what's referred to as a hypotonic solution, that means there are fewer solutes outside than inside. What's going to happen is water molecules on the outside are going to flow into the cell. If I have something that's an isotonic solution, which is the way that we like to keep things in our body, then you're going to have no net movement of water molecules going back and forth. So water molecules are going to go in, water molecules are going to come out, and you're going to maintain this balance, this homeostasis. But if we put a cell in something that is hypertonic, that means there are more solutes outside than inside. In this case, there are more water molecules in here, and so the water is going to move out of the cell. So in this hypotonic solution, it's going to cause the cell to expand, which means it'll actually burst. If we have it over here in this hypertonic solution, the water will leave it, and so it will shrink, and it will actually collapse, which is why it's important to maintain this balance. And so here I've given you a definition of hypertonic and hypotonic. Hypotonic, water goes into the cell from the solution. Okay, this is what if I put saltwater fish into fresh water, then I've got more solutes inside the fish than in the water, and it's going to cause all of the cells of the fish to kind of expand. If I put a freshwater fish into salt water, however, it's going to lose water and it will desiccate. It will actually dry up. And so water will leave the object and go into the solution. So when red blood cells, we have to maintain a certain balance of solutes in our bloodstream in the liquid part of our blood so that our red blood cells maintain a particular shape. If you have too much solute in your bloodstream, your red blood cells will lose water molecules, and so they'll actually shrivel. If you have too little solute inside the liquid of your blood, then the liquid outside your blood cells will move into the blood cells and cause them to expand and possibly rupture. Facilitated diffusion also is something that helps maintain this homeostasis. Facilitated diffusion is passive and no energy is required, but it does have this door. In other words, literally, water moving back and forth across the cell membrane is facilitated diffusion because you have to have these protein channels, these aquaporins, for water to go through. But it's just like an open door. It's not like water simply moves freely around through the aquaporin. It just can't go straight through the cell membrane. Active transport. when we have to use energy to move things into and out of a cell to help maintain this balance. This is called endocytosis and exocytosis. Endocytosis is when we move things into the cell, and exocytosis is when things are moved out of the cell. So that's just a little background about the cell. One of the things that you're going to be asked to do is to take a look at all of the organelles within a cell, and then put down, write for yourself the job of each one of those organelles in a eukaryotic cell. So we'll be doing that in class. All right, that's it for now.